Maggie here. By now you are in your groups and know the food that you'll be working with. The foods that need freeze drying have all been freeze dried for you. This video will show you how we will do your first experiment, which is GCGC MS, and what we gain from this experimental platform. GCGC stands for two-dimensional gas chromatography, and MS stands for mass spectrometry. We use our GCGC MS to detect and identify the volatile compounds that are emitted from food, fresh and while cooking, and which form the aura of the food. Let's look at what exactly is GCGC. Chromatography just means separation. Gas chromatography is the separation of gases using a column. Many compounds of aroma and flavour are volatile, and therefore in the gas phase. Temperature determines volatility of compounds, so some of you will volatilize some compounds by heating your sample to 80 degrees Celsius, and others will heat only to 50 degrees Celsius. This will ensure that the volatiles of interest are emitted by the food. After we collect the volatile compounds from the food, they are injected into a column. The volatile compounds then are looped from this column, and because it is two-dimensional GC, the compounds go through a second column. The chemistry of the second column is different from the first, so the separation of the compounds will be different. Let's have a look at the instrument. Here it is, GCGC MS. Here is the mass spectrometer. The two columns go in here, and there is an oven in here to control the temperature. The samples are placed into the auto sampler, and this is the sample oven that will heat your samples to the appropriate temperature. The auto sampler will place the vial into the oven for 10 minutes, and the injector will then pierce the lid of the vial, collect the gases, and inject the volatile compounds into the columns through an injection port on the top of the instrument. Sample preparation for GCGC MS is very easy. You need to grind your freeze-dried samples and then weigh 1 gram of flour or powder into 2 vials for each sample so that you have replicates. Don't forget to label the vials with the sample number and your group name. Then when that is all done, we will take the vials and load them into the auto sampler. The GCGC takes about 30 minutes per run, and you will be given the data next week in an Excel file containing the names and amounts of the volatile compounds detected. We process that data with a multivariate analysis tool called Principal Components Analysis. PCA does not test any hypothesis. PCA looks at all the variables, which are the metabolites, and look for patterns between samples. The plot then shows groupings based on metabolite patterns, and these groupings are determined by discriminating compounds. We will analyse our data in week 3. Enjoy the prac!